Hello everyone, it's Dr. Rafael Gutierrez again, RH Gutierrez MD, and today I'm actually starting a new project which I'm hoping to help people be able to make models that will help them learn anatomy for anything they want. Now, when I started doing this, I asked students what they needed when they were in anatomy class, and one of the things they always said is that they would want to have models that they could have at home. And so I found a way that you could actually make your own models to help you do studying anatomy at home. But as I started to think about doing this, I also asked my colleagues who teach anatomy and asked them what they would want their students to know before they came to an anatomy class, something that they wish they could recommend students who may be taking a general bio class who are going to be taking anatomy next to understand anatomy a little bit better. The reason I actually started doing this is nationwide I found that anatomy, physiology, and micro, these are three classes, are among the hardest ones for college students. In some cases, I found that a nationwide survey found that about 50% of students who enroll in the class don't finish, don't pass. And so I was hoping to make something that if you are thinking of taking anatomy, you could use it to give you a leg up so you would be able to already have something that will help you succeed. And that's one of the things I did. And what's interesting is as I started asking a lot of my colleagues who teach anatomy about what they would want, they also said something pretty interesting. A lot of them were asked me what I would recommend someone who's teaching anatomy might want to actually focus on in anatomy. The reason is a lot of people who teach anatomy, they know anatomy inside and out, but they don't necessarily know what's important for clinical science. In part because that's not what their job was. They didn't train to be a medical doctor or a nurse. Some of them did actually have done what is it called, chiropractic or even physical therapy. But what do clinicians or nurses actually need to know for anatomy? And when I did that, I actually took a step back and thought to myself, well, what would I want a nurse who's looking at me to be to know? Or if I had a student, what would I want them to know before they went into some sort of clinic? And so I then started breaking it down, and that's actually where this idea for the project came. And one of the things I will do is I am going to use a couple products, and I'm actually going to mention where to get them. You can actually get them a lot of times in different areas, and some of you, especially if you have kids, you may already have this. Now, the first product I'm actually going to tell you may want to get, just to help you out, is one of the 4-color Play-Doh, these things. I actually got blue, red, white, and yellow. And the reason I wanted 4 is so I could actually make different things out of different colors. Because there are structures that are made of certain things, and if you actually get those, you can end up building your own models. And with Play-Doh, if you build it, you could let it dry, and you can reuse them when you're actually reviewing certain things. And so that's actually where I am jumping off, is what is it you would want to know before you even started an anatomy class? And what I would actually tell people is this. People don't take anatomy for the sake of anatomy. Very few people think, hey, I want to learn every bone in the body, every tissue known to man for my own self-improvement. They have a reason to take this. Some of them is for kinesiology, some of it's for nursing, some of it's for medical school, some of it is for their teaching the class. And so I wanted to make something that would actually hit everything and help. And so I actually started with Play-Doh, and I'm not going to actually make all the models myself. I will tell you how to do them so it's quick, but I'm going to tell you how I actually broke stuff down. So for instance, one of the things I decided is if I'm making something that's going to be, let's say, lipid, fats, I would use white. When I actually tell you how to make this, I'm going to draw it on the board so you can see how to make it, and that way you don't have to actually use everything all the time. I also decided that if I was using something that's a phosphate, for instance, which is something that's used a lot of times for structural and for different things, I would use yellow for phosphate. The reason is white for fats, yellow for phosphates. I may actually modify stuff as time goes on, but this is actually a jumping off point. I also decided if I'm making proteins or amino acids, I'd use red. And if I wanted something that was a carbohydrate or sugar, use blue. And so I showed you about the phospholipid. So you have a phosphate and a lipid. And so you have two lipid tails and a phosphate head. That's pretty much the basic, as how far I'm going to go into the basic structure of a lot of things. I am going to tell you about the organelles, little organs. And the reason that's important is how it actually breaks apart, how it actually changes depending on what organ system you're studying. Now, if I was actually doing a glycoprotein, a protein with a sugar on it, we got a protein, it could be an enzyme, and if I wanted to make it a glycoprotein, sugar protein, there you got a glycoprotein. If I wanted to do a 3-chain amino acids with 3 phosphates, or 1 phosphate, I could actually have an amino acid. The protein is what I would use, and a phosphate, and you would have a, let's say, adenosine monophosphate, mono for 1. And that right there is going to tell you something. There are words that mean things in anatomy. So lipid, or lip, is always going to mean fat. So if you hear of something called a lipolysis, lip or lip is fat, polysis, breaking. So lipolysis is breaking fat. Glycolysis, breaking sugar. You also do have a lot of other words which I'm going to mention later. But I'm not going to actually use the Play-Doh for myself because I don't want to actually deal with all the structures in all the cell and give you. I am going to give you the phospholipids, which is a phosphate and 2 lipids, and the proteins, which will actually make sense later. Now, I'm going to use my whiteboard for the first part. And instead of using white for phospholipids, I'm right now going to use black. And the reason I'm using black for lipids is because white won't show up on a whiteboard, right? So I'm getting my whiteboard, and here it is. If we're dealing with anatomy, we actually do want to talk about a lot of different things. First is the basic unit of life. And the basic unit of life is the cell. And if you know the cell, you actually have a big portion of what you're going to be looking at. So body starts actually with cells. From cells, it goes to tissues. And a lot of people get involved in tissues. And I'm only really deep into tissues. And what I'm going to do is, to make it easier and faster, I'm only going to talk about tissues that are specifically needed for the organ system I'm talking about. So I'm not going to tell you all the tissues all at once. I'm going to tell you these are the tissues we're dealing with. Now, one of the things that happens is a lot of tissues interplay with a lot of different things. A lot of organs, tissues come together to form organs. A lot of organs play with other organs, so you have organ systems. And sometimes you have organ systems which interact, actually a lot of times, most of the time, you have organ systems that interact. And so what I'm going to do is I'm going to build up. So if initially I'm going to talk about the skeletal system, and you're going to see how all the skeletal system comes together with the tissues and how it's formed, which will help you understand why it's a certain way. Then I'll go into other aspects. So if I go into skeletal, I'll use the tissues that are found in bone and the skeletal musculoskeletal system and put them together. So you'll actually already have some, and we'll have just a couple more. And so on. It's just going to build up on itself. It may sound a little intimidating initially. It will make sense. And so if we're actually talking about the cell, and like I said, if you're making a cell model, use the whites for fats, the yellow for phosphates, blue for sugars, and red for amino acids. And amino acids build up proteins. So if we actually start with the cell to begin with, I know I made a big video on cells, but someone asked me to make it simple if I could. So if we start with cells, we would actually have a phospholipid bilayer. Phospho for phosphates and lipids for fat. So phospholipid bilayer would be you would have the 2 lipids coming off, and you'd have the phosphate on top, right? So, and it's a bilayer, so you have 2. So this would have one here and one here. You can see my phospholipid. And once you have your phospholipid, you have your phospholipid bilayer. Now, I'm not going to draw the bilayer like that. I'm just going to draw it as a single line of black. If you're making a model, your phospholipid bilayer would be just the white lipids. And so you'd have a big, really big bag, which is your cell. Now you may not be able to see it too much right now, but you'd have a big bag, which is your cell. Your phospholipid bilayer would be on the outside. And you have another lipid in here. And that lipid in here is your nucleus, which holds your DNA. If I really wanted to do DNA properly, I would get my sugars, which I told you I'm using blue, so blue. And I would also use, so you'd have your sugars here, and I would also use my phosphates. I'm sorry, my phosphates, which are yellow. As I don't have yellow ink and it wouldn't show up, I'm going to use green for the phosphates, and it would end up making the two structures here. So you have your genetic material inside the nucleus. The nucleus is a phospholipid bilayer, as is the outside layer of the cell membrane. And I'm going to give you one of the first words that will help you understand anatomy and pretty much all types of cell biology. And one of the first words I would actually give you is cyte. C-Y-T-E. Cyte. And my black is running out, so I'll switch to purple. So cyte technically means bag. But when we're dealing with cells, cyte means cell. So we have a phospholipid bilayer, and the fluid inside here, inside the cell, would be your cytoplasm. Plasma is fluid. So cytoplasma is a fluid inside the cell. You have your nucleus. Now, you have some protein catalysts, which are called enzymes. And you have enzymes scattered throughout the cytoplasm. I'm just going to put a bunch of dots for the proteins that are in there. You also have proteins that make tunnels through the membrane here. This is called the nucleus, so that protein that makes a tunnel there is a nuclear pore. And the way it works is you do have an enzyme inside the nucleus, which is called the enzyme portion that I actually would tell you to know first is called a transcriptase. And transcriptase, anytime you see ACE, A-S-E, it's telling you that it's an enzyme. Enzyme is just a protein catalyst, and a protein catalyst is a protein that causes things to happen. They don't technically cause a thing to happen, to be honest. It speeds up something that's going to happen. Makes it easier to happen. And one of the transcriptase, sorry, is going to take the DNA and make it into RNA. And the RNA is going to leave the nucleus. So I'm not going to try to draw everything again. I'm just going to draw the RNA as a little green thing that leaves the nucleus. And so it's going to be out here. So now you have your RNA molecule there. And your RNA molecule meets a specialized enzyme which has two parts, which is called a ribosome. And a ribosome, what it does is it makes proteins. So it's going to take your RNA and make proteins. If the protein is made in here, one of the things that these proteins can do is give your cell structure or a cell skeleton. If you remember, a cell is a site, and so we call it a cytoskeleton. Cytoskeleton would be the protein structure inside the cell. That's going to be very important in certain types of tissue, which I'll tell you about later. Now, sometimes you want to make a protein like the one here in the phospholipid bilayer around the nucleus, and so you would make it in a slightly different place. What you would make it in, I'm going to use purple again for the phospholipids now. It would be a membrane coming off of this, going all over the place, like that. So if you're making the cell, you'd make a white circle for the cell membrane, another white circle for the nucleus. You could put a couple proteins in there, so the protein to give you a nuclear pore. And then you have a phospholipid bilayer in here, which is called an endoplasmic reticulum. And you can actually make this with the Play-Doh, like I said. Now, the endoplasmic reticulum has two types. You have smooth, which usually stores and releases calcium, breaks down fats, makes fats. We'll talk about that later. And you have another one, which is called the rough endoplasmic reticulum. And the reason it's rough is because it has ribosomes. And I'll tell you why that's important in a couple seconds. So we're going to put a couple figure eights here. Again, if you're making this phospholipid bilayer, you'd make it with your whites. The ribosomes, you would actually make it with your bread. See, there you go. 2 little subunits for ribosomes. And you can just put it on your phospholipid bilayer. Now, what usually happens is the RNA molecule, if it's going to be for protein inside the cell, it goes to the free ribosome. If it has to be bound to a membrane, or if it has to go into a little sac, it'll be made on the rough endoplasmic reticulum. Smooth, no ribosomes. Rough has ribosomes. And so you make your proteins here on the rough endoplasmic reticulum, so you can put it in a little bag. Now that bag would be made from phospholipid bilayers, from phospholipids, and it would be a bilayer, and you'd end up putting the protein in a little bag in here. So now this bag would actually butt off and be in the cytoplasm, and the protein that you made from here would actually be in a little bag here. Or it could even be attached to the phospholipid bilayer. So you can actually make the nuclear pores by doing this and bringing it back to the nucleus. It'll actually be fused. Now this is a vacuole, and there's a lot of different types of vacuoles. This is a vacuole here. And vacuoles can sometimes be sent out, but before they're sent out, you have to decide where it's going to go. And for you to know where it's going to go, you need a sorting center. And that sorting center is another structure in here. Usually people will say it is a phospholipid bilayer as well, and it's called the Golgi. I'm right-handed. The Golgi. So you have the endoplasmic reticulum, rough, and the vacuole here. I'll put V for vacuole. And it's going to go from the rough endoplasmic reticulum to the vacuole, inside the vacuole to the Golgi apparatus. And the Golgi will decide if it has to go to the nucleus, or if that vacuole will be sent somewhere else. It can be sent to fuse with the cell membrane. And that's going to actually give us another word that's actually important, especially when we start talking about skeletal system. There's something called exocytosis. Anytime you see the word exo, it means outside. So exocytosis is outside the cell. Osis is processing. So you can actually take the Golgi, and you can take that Golgi and send it out of the cell. And the way it actually goes out is the phospholipid bilayer around that makes the outer portion of the vacuole will fuse with the cell membrane. And so when it fuses, what you end up having is this comes together, and so whatever's inside the vacuole will be pushed out. And that process is called exocytosis. So if you're making a protein to be sent out of the cell into, let's say, the extracellular area, you would do that through exocytosis. And that's all I'm going to talk about today, because next video, I'm really going to talk a lot more about the compact skeletal tissue, something called the osteon. And if you understand the cell, you'll understand why embryology happens a certain way and why the structure happens a certain way. I hope you enjoy this and have a nice day.